So thank you very much, and I will give you some background on the genetics of autism for those who don't know, and some news about the new results in the lab. So my lab is mostly working on the social brain, and as David said, I think, and as a geneticist, I think genes don't care at all about the DSM-4, DSM-5. They never read it, and I think that this silo with autism, and that's the idea to have more dimensional is super important. Also, super important is to work with different view of what we call autism or the social brain. So I always ask, can you see the psychiatrist? So there is one psychiatrist here, also here, a psychologist, neurobiologist, someone listening to mice, and some geneticists, and working at the same cafe machine at the Institut Pasteur, working on the social brain. So this is the autisms, but always super different from one child to another, or from one adult to another. As you know, there is two diagnostic criteria now from the DSM-5, lack of social interaction, stereotypy. The language problem is not anymore in the DSM-5, because some people with autism, they speak, and some people, they don't. We still don't know why there are more males than females in autism. What we know is that we are very bad in diagnosing female in autism, probably. And that's why we have more males than female. And that probably female, there's a camouflage or a buffering that they have a different kind of autism that we have to understand why. It's always autism plus. It's always autism plus something, as David said, with intellectual disability, epilepsy, and anxiety, which is super important in the outcome of the person. So the prevalence is about more than 1%, I will say. I have only 2 slides on this. There is an increase of the prevalence of autism, as you may know. So it was 1 in 5,000 in the 70s or 80s, and now it's 1 in 40 in US. When you look, in fact, in the, this was a work by Christopher Gilberg in Sweden. When you look at the autistic traits in the population, they don't really go up. But indeed, the number of people with autism diagnosed in the world, at least in Sweden, is increasing. And I had the chance to listen to Stephen Shore. He's a person with Asperger's syndrome. He wrote the book Autism for Dummies. And he gave a really great seminar and said that the autism is increasing just like the number of FedEx package in the world. And he said that probably there is no correlation between FedEx package and autism, which you never know. But what he said to us is, can you tell me what is between the E and the X in FedEx? So there is an arrow. So so every time you will see a FedEx package, now you will see the arrow. So two messages, I think. I think Stephen Shore is right. I think the autism is a little bit like the arrow of FedEx, because it was there, but you didn't know it was there. Now you know it, and then you see it. So it's really the medical awareness that is one of the great part of the variance. And the second message is, I think we have to be careful when we say cure autism. I think I don't want to cure this person. I think that's another discussion we have to. As a geneticist, super important. Okay. A second person with autism that I like the way she's illustrates she said that it's funny when I meet medical doctor or researcher, they see my life like this, and it's funny because I see my life more like this. And I think that's it's interesting. So we clearly see them as gene A or one gene and autism, and we know it's much more complex than this. Okay. So what are the progress in the genetics of autism? So in the 80s, in 1985, when I was 20 years old, there was about 1% of only chromosomal abnormalities, and then with the genome, exome, and so we have now about 10 to 20% of genetic cause. We think we can diagnose a genetic cause of autism in about 10 to 20%. As David said, I think that's super important that the 10 to 20% is mostly patients with autism plus intellectual disability that we are this yield of diagnostic cause of autism. So when you look at the heritability, we're very bad because in intellectual deficiency, they have about 40% of diagnostic yield, and in epilepsy, even more than 40%. So I think why it's so complex. First, we start later than intellectual disability and epilepsy, so we have to work more. But also, it's super complex in terms of genetic architecture. So you have this kind of situation where the father is okay, the mother is okay, and then the child has a mutation, and you have like a de novo mutation, you have autism. And on the extreme right here, the parents, they probably have a lot of risk alleles, and they additionally converge into the child, and then it's through a threshold, and you have autism. So in my lab, I said I have my monogenic friends who think that autism is like this, and my polygenic friends who don't care at all about this, but they care about the p-values and the common. So they are in the same lab and talking together. So from this monogenic, more than 15 years ago, we found this neuroligin mutation on the X chromosome, then SHANK3, and then neurexin and other genes. And so these are the genes that are found. It's about 5 years ago that we made this review. So it's on chromosome 1, 2, 3. And so as you can see, only in France, people think that there are still no gene for autism, but it's a lot of genes. And I would say it's super heterogeneous, but it's likely we have some pathway that are, they converge into some pathway, and this is one of the protein-protein interaction network at the synapse. So there are two main pathways. I will not go too much in details. One is the synaptic pathway, and one is the chromatin remodeling pathway, which is really interesting. In the second part of this genetic architecture, so this one is the black hole. I should have put the new picture of the black hole. It's my, and we are working on this. But this is interesting. So this is the common variant. So how do you find this common variant? You have more than 16,000 people with autism, so it's super categoric. You have autism or not, which is okay. But it's okay. I mean, we can find some. The last paper, it was 18,000 people with autism, and only 5 genome-wide significant SNPs. So why so? This is super important because these common variants are really important but difficult to handle. We know there are many variants that are involved, but each one has a super small effect. And this is super well illustrated in the last review by Pat Sullivan and Dan Geschwin in Cell. So this is the genome-wide significant loci, okay? And this is the heritability. So the heritability for schizophrenia and autism is about the same. But you can see that we are here. We have about 5 genome-wide significant loci, and they have about more than 100 and more than 200, but they have more patients. So this is really like the curve is going more and more and more, and we will get this common variance. But it's because it's small effect, we need a lot of patients. So at the end, there is now a Spark gene list. If you're interested, you can go to their website. well curated by medical doctors who say that for these genes, we can go back to the patients. I mean, I can talk more about it. The SPAC, there will be 50,000 people in US collected, and they will go on genetic tests, exome on saliva. And on the common variants, they are really important but difficult to handle, again, from one patient to the other. So what we are doing is this Polish-NIC score, and to aggregate the variants and to see if there is something. And for example, the 10th percentile with the highest Polish-NIC score, they have a odd ratio of 3, which start to be interesting. So what's new from my lab? First, we are creating new tools to handle this complexity. We met Gravity. I don't know if Sébastien is there, but he will probably work on the CNV, talk about the CNV. We are working a lot on the polygenic, and we are following the genes on Shankopathy. So Gravity is one tool. You can go to our website. I'm here for the whole day, so you can talk to me. This is this is a tool. This is a Cytoscape plugin. Normally, you see this. You have your favorite gene that you think it's super important, and then you have the mutation, and you say, okay, I found. But in fact, if you look at the mutations, we have about 3 million of variants between you and me, about 10,000, more than 10,000 in the exome. And in fact, this child has this mutation, but also this one, this one, this one, this one, this one. So it's a way to visualize the pathways and where the mutations are coming from the father, coming from the mother. You can go, and we have a new VCF version for the aficionados of the genome and exome. Okay, the polygenic. So what we asked, we are contributing to this autism versus control, which I think is interesting, but again, I like more dimensions. And so with Simon Baron-Cohen, we recently met this, the read the mind in the eye test is a very standard test for empathy or cognition. You just have the eyes, and you have to say if you think this person is playful, comforting, irritated, or bored. And then you click, and then you have many eyes, 46 pairs of eyes. And if you have a score in that part, you're okay, and if you're there, it's okay, but you're probably not very skilled to see the read the mind in the eye test. So my brother-in-law is there, and I like him. He's voting Trump, but it's okay. So you have these scores, and then you can ask to twins what is your score, and we could see that 28%, which is of the variance, is explained by the genetic difference. And then we put I could be in jail in France because of this. We put this test on the 23andMe website, and we get 88,000 responses. And then we could calculate the heritability looking at the SNPs. We could find 6%, highly significant, but only 6% captured by the SNPs. And then you can do a GWAS. And so we found the first. There is 88,000 people and only one genome-wide significance, only in girls. And so recently, and we made a questionnaire with Simon Baron-Cohen, the empathy questionnaire and the systemizing questionnaire. I mean, everything is almost published. The empathy quotient, we get this. We made a GWAS on empathy. It was only 50,000 people. And on systemizing, so the way we like to solve problems, this is not, this is in bioRxiv, but it's the paper. So you can look at the GWAS of systemizing, and then you can see if the SNPs that are involved in systemizing are also correlated with other aspects. And so this, I will not go back, but the most interesting, I think, results from this paper is that we looked at the empathy, we looked at the systemizing quotient, and then we looked with the GWAS from other traits, what was correlated. And you can see here in autism that the empathy, so the SNPs, the variants, genetic variants, that give you a low score in empathy are there, I mean, the SNPs are there in the autism genome. The SNPs that give you a high systemizing are there in the autism GWAS. So then we can start to see what is the contribution in the autism susceptibility genome, what is the contribution of empathy and systemizing. And I think that that's one of the future, instead of looking only at autism and yes or no. Okay, so that's the polygenic. We have also a recent paper that just 5 days ago in bioRxiv. 4 years ago, we looked at the heritability of the brain. So looking at brain MRI of individuals from the Imagen cohort. And we looked at and it was only 2,000 adolescents with MRI. Now we get 20,000 participants because we downloaded all the biobank brain. And so the heritability was, as you can see, it was about 50%, but the error bars were super high. And now we are very happy because about the same results, 50% or 40% of heritability, but the bars are very small, the p-values are. So the more it's really important because the common variants are not, at least in France, not very well studied. And the more you add SNPs, the more you have heritability. So you have more SNPs, and you have more heritability. The SNPs are probably mostly in the gene. And we met this, the part of your brains are correlated in your brain, the size of the volumes of the organs, of the part of your brains are correlated. And they also are super genetically correlated. So something that makes a high hippocampus is also correlated to the high brain volume, for example. So from this data, can we estimate with just a SNP array what will be the size of the brain of someone? And I will say yes and no. I mean, to date, no. So what we have done is to take the SNPs and then to use 6,000 people that were not part of the GWAS, and then to make the polygenic score and to see what's going on. So this is a result. So if you like p-value, you're super happy because it's super significant. But this is the people with a low polygenic score for a small brain and a high polygenic score for a bigger brain. And you can see that, again, if you like p-value, you're happy. But you can see that the overlap is really high. So can we do better? Probably. And we looked at the height. Okay, it's super simple. It's phenotype. We looked at when you take the 13,000 people from UK Biobank and you try to estimate the 6,000. Again, it's super significant, but the overlap is quite high. When now you take the 250,000 people from giant GWAS and you have better p-values, then you start to see that people with low GPS, polygenic score for height, are indeed really smaller than people with a high GPS when they're taller. So you can use this in the clinic. Maybe one day you will have this polygenic score to interpret the susceptibility to autism or to anything. Okay, so really last the Schenk the Schenk story. And more than 10 years ago, we found this mutation in autism, and after Claire found that a Schenk2 mutation, Schenk1 mutation, and this is the IQ, and this is the autism diagnostic interview. And you can see that patients with Schenk1, they're very high, normal IQ. Schenk2, they are intermediate, and Schenk3, they have lower IQ. So we can do this kind of genotype-phenotype correlation. So in mice, what we have shown is that a long time ago, we found that these mice with Schenk2, they have hyperactivity. That's what I say. If a mouse is going to a clinic, you will go directly to the ADHD specialist. And so this is and so we try to cured the mouse with methylphenidate. It was a complete failure. They were even more hyperactive. And so I was depressed, but then I realized that it was a good model to test other drugs. And lithium, by the way, is making them better. The social interaction, so this is published for a long time. We found that this mice, this is a male mouse, and we will put the female mouse, so this is what's happening. And this is the SHINE2 knockout. So you can see the different, so everything is public. You can go to there are stats, and but some people ask me if it's because he doesn't like girls, or and in fact, so we have tested two things. The first things we've tested girls only, and we tested if they were grouped or isolated. And I think it was interesting. So we keep the mouse grouped and or isolated for 3 days, and then we take one mouse and we put it together with another mouse. And that was interesting because if you group the mouse, then you quantify the time in contact with the new mouse. They go to the mouse sometime not. But when you isolate them for 3 days, they're quite interested, more interested by the other than if you group the mouse together. And then for the SHINE2, you can see that they're not super interested, but not a lot of contact. And the fact that you have isolated them was not a super trigger to go to the others. So like they're not really, it's not maybe social reward, but something like that. Then you can record the mice, and to listen to the mice, we have a lot of stuff like this. So again, if you quantify the vocalization of the mouse, when you group the mouse, they vocalize sometime, but when you isolate them for 3 days, they're vocalizing to the others, and the SHINE2, they are not really vocalizing. And again, so you see this one vocalize, so she can vocalize, but she doesn't. I mean, looks like most of the time they don't care about the other. Okay. I said to Elodie, if you want to understand what these mice are saying, you need something like a lot of vocalization, like YouTube for mice. So something like MouseTube. So normally when I say things like this, people don't care, but she cares. She's really nice. And she made MouseTube, so it's a platform of data sharing of mouse vocalization, if you want to go. So she's an ethologist. She was listening to mice, to baboons in Nigeria, and 10 years ago. So when to take the mouse and to test the mouse for 2 minutes, she thought it was awful. So she wanted to have something that is long-term, more ethologic. So what she has done is this. It's really amazing. So we have the first tracking system. So each mouse is tracked. We know exactly what they're doing. You can even, it's not camera on the mouse. Huh? It's just the image. And so you can see what the mouse is doing. We have the ethogram. This is a SHINE2 wild type behavior. I will not go too much in detail because I'm super late. This is my Andy Warhol way of. But for each mouse, you can see what they are doing. If you're interested, if there are some mouse people in the lab, in the audience, we made a IKEA type of to construct the system. We made a movie, and the paper is in June, and it's already 18 labs. It's not in a regular paper. It will be soon in Nature Medical Bioengineering, but it's interesting. Okay, humans. I will go very quick. We have patients with SHINE3. We made iPS cells. It's a long work. We found that they have very low mRNA in SHINE3. And the surprise that even if they have low mRNA in these glutamatergic neurons, they still have the mutation. So for the aficionados of the mRNA in the room, it looks like they have lower mRNA, and the lower part is, half of them is totally non-functional. At the synaptic level, there is a clearly decrease of synaptic density in 4 patients with SHINE3 mutation. And when we have done the RNA-seq, we can see that it confirmed the low SHINE3 mutation, but also there was also a lower number of FMRP target genes. So probably you have a problem with SHANK3, you have a problem in synapse, and then you have more problems than only SHANK3. Okay. Then we use these cells to make a screening of 205 FDA-approved molecules, and we found that lithium was one of the molecules that was increasing the SHANK3 mRNA. So we are starting now a clinical trial on lithium for the patients with SHANK3. Okay, that's the what's next. So as David said, that it's very important to, if you see one patient, it's very difficult for the causality of, I mean, if this variant is really involved, I mean, you need a lot of network to understand what's if you see the same patient with the same kind of mutation. So we, with the EUMs or the European level, we are looking at different cohorts from different time, and we are using a lot of battery to, instead of saying autism, to say something more dimensional. So that's the end. It's like a synaptic orchestra. I can see this now with SHANK3 here. So if you remove this person, probably the song will be different. If you remove one guy here, if you have a good ear, you can make a difference. But if you remove all these guys, then probably there will be a difference. So these common rare variants. I will have not time to talk about resilience, but we are working to try to understand when you have a strong mutation, some people are not sick, and we want to understand why. I love, she was one of my heroes. If you have seen One Child with Autism, you have seen One Child with Autism. And I couldn't resist. This is another hero that I will see tonight. That's why I'm super. I always love that quote. I accept chaos. I'm not sure whether it accepts me. And I think if you work on genetics of psychiatry, you need to accept chaos. Okay. The dream team, wonderful people working in the lab. L'argent, super important. And the families. And I love this father who found he made a really beautiful comic book and a YouTube movie on the autism of his daughter, and on the T-shirt it's written, "I am unique just like everyone else." As a geneticist, this is my message, and merci beaucoup.